Dear student, I am Dr. 𝗑𝐲𝐫𝐞, Professor of Biochemistry. So, in this lecture on special products coming from amino acids, I will be going over three special products, that is creatine, carnitine and polyamines. Let's get into the details. First is a creatine phosphate. Creatine phosphate, it is coming from two amino acids, that is arginine and glycine. Note that in the kidney, arginine is going to condense with glycine by amidotransferase enzyme and form guanidinoacetate. So, the guanidinoacetate formation happens in the kidney. Now, this guanidinoacetate is taken to the liver, and in the liver, guanidinoacetate undergoes methylation reaction catalyzed by methyltransferase enzyme using SAM as a methyl group donor. That is 𝗠𝐴𝐵𝐚ethionine, which is the derivative of methionine. It acts as a methyl group donor for the conversion of guanidinoacetate into creatine. Now, once creatine is synthesized in the liver, this creatine is secreted out into the blood, which are taken up by the muscles. And in the muscles, under resting condition, there will be phosphorylation of creatine to make creatine phosphate. And the creatine phosphate is a high energy molecule, and it is going to store a readily available energy for future needs whenever muscle needs ATP to be made from ADP. And that's how the creatine phosphate is stored. So, when the muscle contraction happens, see the readily stored energy in the form of creatine phosphate, it is undergoing a dephosphorylation process by breaking that energy rich phosphate and becoming creatinine molecule. And while this process is going on, so the ADP will be phosphorylated to become ATP. So, ATP can be generated by breaking creatine phosphate. So, if that ATP can go into metabolic reactions. Now, the product that came out is creatinine, and creatinine is a waste product and it is secreted out of our body in the urine, and that is done by the kidney. So, the blood levels of creatinine remains constant unless there is any problems with the kidney, including renal failure. And also the creatinine levels in the blood is measured as a useful marker of kidney function test, because unlike blood urea level, serum creatinine levels do not undergo physiological variation. So, as I explained in earlier slides, creatine phosphate is the energy source in the first few seconds of muscle contraction by breaking creatine phosphate here. During sustained muscle contraction, glycolysis, especially the aerobic glycolysis or oxidative metabolism, can go on, and later when the oxygen deficiency occurs or when there is a limitation in the oxygen supply, at that time glycolysis will switch to anaerobic. And also creatine phosphate, it accounts for 1% of the weight of skeletal muscle, and the quantity of creatine is proportional to the muscle mass. That means a person having higher muscle mass will have higher creatinine levels, because they will simply have higher creatine phosphate, which will break into creatinine when muscles undergoes contraction. So, when the muscle mass decreases, like in muscle atrophy or say Duchenne's muscular dystrophy, so creatinine levels will decrease. So, increased presence of muscle specific creatine kinase in the blood is an indicative of muscle damage, because creatine kinase, what it does, it is going to convert creatine into creatine phosphate. So, higher the activity of creatine kinase, so that means muscles are active, and also higher presence of creatine kinase in the blood indicate that there is a damage to the skeletal muscle, and the things that are there in the skeletal muscle are simply excreted. So elevation of muscle specific creatine kinase, and that is 𝐒𝐾MM, it indicates damage to the muscle. Now, when a creatine phosphate undergoes degradation, the phosphorylasis, where the phosphate is separated from creatine, so the product that forms is creatinine, and this creatinine is excreted in the urine. And the amount of creatinine that is excreted is proportional to the total creatine phosphate content of the body. So that means when the muscle mass decreased, urinary creatinine levels will also fall. So measurement of serum creatinine is useful as a kidney function test, because compared to urea, serum creatinine do not have a physiological variation. And the rise in blood creatinine is a sensitive indicator of kidney malfunction. So these are some of the points related with creatine phosphate. Going to carnitine. Carnitine is a dipeptide synthesized predominantly in the liver by using methionine and lysine. And in the carnitine biosynthesis, there is an involvement of an enzyme called gamma-butyrobetane hydroxylase. And this gamma-butyrobetane hydroxylase is inhibited by a drug which is called as meldonium. Meldonium, as I explained to you in carnitine shuttle mechanism during fatty acid oxidation process, meldonium is a doping agent abused by a sports person, because meldonium, it is a modulator of L-carnitine, thereby it is going to modulate our metabolism from highly aerobic metabolism, which is oxidation of fatty acids, to oxidation of glucose, thereby decreases the oxygen demand by our tissues and thereby improves the muscle function, including the athletic performance. So this is what happens, and carnitine as such, it is involved in the transport of long chain fatty acids across the inner mitochondrial membrane. Now coming to polyamines. Polyamines are the other special products that are coming from amino acids. So polyamines, as the name says, they contain multiple amino groups. Basically, important polyamines are putrescine, spermidine and spermine. So ornithine, which is a product that comes out of breakdown of arginine into urea and ornithine in urea cycle, this ornithine can undergo ornithine decarboxylation reaction to make a putrescine, and the putrescine will condense with a decarboxylated product of 𝗂𝐵𝐴𝐷𝐻𝐿-methionine, that is 𝗂𝐵𝐴𝐷𝐻𝐿-methylpropylamine, and this decarboxylated product of 𝗂𝐵𝐴𝐷𝐻𝐿-methionine, when it combines with putrescine, it is going to make spermidine, and spermidine is going to condense with one more decarboxylated product of 𝗂𝐵𝐴𝐷𝐻𝐿-methionine to make spermine. So we have putrescine, spermidine and spermine, which are referred as polyamines. And these polyamines, because they have a basic in nature, that means they have got higher positive charges, so they readily interact with the negatively charged biomolecules like nucleic acids and membrane phospholipids. So their interaction with the DNA and RNA results in cellular changes, and also their interaction over cell membrane may enhance transport of specific substances across the membrane. So, and also they may play a role in hormone mediated actions on membrane fusion during exocytosis and endocytosis, and certain enzymes are also inhibited by polyamines. Example, protein kinase enzymes. So these are all special products coming from amino acids. In that, I have went into the details on creatine, carnitine and polyamines. Thank you.